Welcome everyone to our Insights in Vincent Forum, profiling the importance of early career cancer research and our St. Vincent's partnership with Cure Cancer. Before we begin, I'd like to acknowledge the Gadigal people of the Eora Nation, the traditional owners and custodians of the land on which we meet, and pay my respect to elders past, present and emerging. As many of you know, I'm Richard Haddock, the Chairman of the Board of St. Vincent's Current Foundation, and I am delighted to be with you. I'd also like to acknowledge that we are joined today by two sisters of charity, Sister Margaret Fitzgerald and Sister Anthony Groves, my fellow St. Vincent's Current Foundation board members and St. Vincent's colleagues, Toby Hall, the Group CEO of St. Vincent's Health Australia, Charles Curran AC, the Chairman of Trustees of the St. Vincent's Current Foundation, Margaret Cunningham of the J.W.M. Cunningham Foundation, and special acknowledgement to Miss Nikki Kinlock, the CEO of Cure Cancer, who's also joining us on our panel today. The St. Vincent's Current Foundation works in partnership with many other foundations and fundraising organisations, including Cure Cancer, as part of our efforts to support our St. Vincent's hospital priorities. One of the key priorities is the development of our doctors, nurses, and clinical researchers for the future. Another is the acceleration of translational medical research into new treatments and advancement in patient care across the Kinghorn Cancer Centre and St. Vincent's hospitals. St. Vincent's and Cure Cancer are committed to supporting early career cancer researchers. In 2020, we announced a 3-year joint funding partnership between our two institutions that is today supporting immunotherapy research for lung cancer. The partnership has enabled the work of Dr. Vanessa Chin, whom you will hear from today, which is contributing to an important body of knowledge in the application of immunotherapy, as well as fostering our next generation of medical leaders. November is Lung Cancer Awareness Month, and today we are going to share with you our efforts to fast-track immunotherapy research that will pave the way for personalised treatment options and improve outcomes for patients using lung cancer. I would like to extend my thanks to Cure Cancer and to all of our donors that are supporting Dr. Chin's cancer research here at St. Vincent's, including Mr. and Mrs. Avrom and Lindy Sank, and Mr. Theo Theodosi, a donor and St. Vincent's patient whom you'll hear from today. Special thanks also to the Vidal family and Ron and Robin Finnemore, whose visionary support enabled St. Vincent's Hospital to acquire a state-of-the-art lung CT scanner in May 2020, which today is providing superior diagnostic capabilities for the benefit of lung patients, including those with lung cancer. This support has had even greater significance than we would ever have imagined as our hospitals have responded to the COVID-19 pandemic. And I'd also like to acknowledge the long-standing support of the Kinghorn family, who helped establish the Kinghorn Cancer Centre at the St. Vincent Darlinghurst precinct. I'd now like to hand over to our expert facilitator, Mary Lloyd, who will introduce our panel and guide the discussion. Mary, who's joining us from South Africa today, worked as a journalist in Africa, the US and Asia before moving to Australia in 2009. She was the business producer for CNN in Hong Kong and has reported and produced for the South China Morning Post, Agence France Presse, NPR, and Channel 9. With the ABC, she has reported on federal politics from Parliament House, covered foreign news on the international desk, and now works in the specialist reporting team. In 2020, she was treated for breast cancer at St. Vincent's and now describes herself as a cancer survivor. Welcome, Mary.
Thank you so much, Richard. It's such a privilege to be involved in an event like this. As you said, I went through cancer treatment last year, and while I'm enormously grateful for the therapy that I was offered, and I'm determined to be nothing but convinced that it worked. I think, like many cancer patients know, that fear of the dreadful stuff never really goes away. But one thing that most certainly brings me great comfort is knowing that right in Sydney, at the very place where I was treated, world-class research is underway to move cancer therapies ahead. So I'm delighted for us today to be discussing how the partnership between St. Vincent's and Cure Cancer is progressing. Our panelists are here today to share their thoughts on the importance of early career funding for cancer research. And we will get a first-hand account of the transformational impact that this can have for cancer patients and their families. As Richard indicated, this partnership has kicked off by providing funding support for immunotherapy research for lung cancer. Now, in Australia, lung cancer is the 5th most common cancer diagnosed, excluding non-melanoma cancers. It is the leading cause of cancer death and responsible for almost 1 in 5 of those deaths. Fortunately, immunotherapy is making great strides towards advancing the management of lung cancer. This type of treatment is an antibody therapy. It stimulates the body's immune system to fight the cancer. And in doing so, it offers a great deal of hope for patients. However, much is still unknown about why the treatment works so successfully for some and not for others. To unravel some of this for us today, let me now introduce our panel. Professor Anthony Joshua is the head of medical oncology at St. Vincent's Hospital in Sydney. He specialises in genitourinary malignancy and melanoma, with interests in immuno-oncology and skin cancer. Try and say that fast. Anthony is also responsible for leading the medical oncology program at the Kinghorn Cancer Centre. And as well as being a place that provides exceptional clinical care, the Kinghorn Cancer Centre is driving a lot of cancer research. It does so by working closely with the Garvan Institute of Medical Research, which now has 30 years of world-class cancer research behind it. It also leans on the 150 years of specialist compassionate cancer care that's been offered to the community at St. Vincent's Hospital. The Kinghorn Cancer Centre, or as I fondly refer to it, the Kinghorn Day Spa, places the patient at the centre of all decisions. One of the ways clinicians achieve this is by rapidly translating the research findings into new approaches for personalised cancer diagnosis, treatment, and prevention. Welcome, Anthony. Now, someone who knows all about translating those research findings into clinical care is Dr. Vanessa Chin. She's a medical oncologist and postdoctoral researcher working at St. Vincent's in the Kinghorn Cancer Centre. She's focused on the fields of lung and head and neck cancer. During her PhD, she worked with the Personalised Cancer Therapeutics Group, which looked at using genomics to inform therapeutic discoveries in pancreatic cancer. Her work included a meta-analysis, in vitro and in vivo analysis of rationally selected drug targets, and analysis of metastases forming in real time. Vanessa is based at the Garvan Wiseman Centre for Cellular Genomics, where she is the Lung Cancer Research Fellow. Her work is now being funded through the partnership that we're here to discuss today of Cure Cancer and St. Vincent's Curran Foundation. Her research is examining the genes of cancerous and non-cancerous cells from patients' lungs to see whether it's possible to personalise treatments for them and to help predict which patients are most likely to benefit from immunotherapy. It's great to have you here today, Vanessa. Also joining us is Nikki Kinnoch, who is the CEO of Cure Cancer, which is dedicated to funding cutting-edge research across all cancer types. To that end, Cure Cancer gives talented young scientists the opportunity to make an impact with their innovative and groundbreaking ideas. They are the only Australian organisation dedicated exclusively to funding emerging researchers, creating that pipeline of future research success. Cure Cancer seed funding has kickstarted the careers of over 534 cancer researchers and led to numerous global breakthroughs in brain cancer, breast cancer, immunotherapy, pancreatic cancer, childhood leukemia, and more. It's an impressive list. Welcome, Nikki. And we're also very pleased to have with us today Mr. Theo Theodosi, who is a grateful patient of and generous donor to St. Vincent's Hospital. Diagnosed with stage 3 lung cancer in 2017, he wasn't a suitable candidate for surgery, chemotherapy, or radiation because the cancer had appeared in his lymph nodes. Now, under Dr. Chin's care, he started a program of immunotherapy, which resulted in complete resolution of the cancer in his lymph nodes and paved the way for successful surgical removal of the remaining cancer. It's wonderful to have you here today, Theo. Anthony, let's come to you first. The landscape of cancer research is often difficult for non-medical people to navigate. Maybe you could start by giving us a bird's eye view of what the priorities for cancer research at St. Vincent's are and the overall approach that you're leading.
Thank you very much, Mary. So our vision at St. Vincent's at the Kinghorn Cancer Centre is to really realise the promise of innovative, equitable and personalised medicine and medical care, cancer care for all patients affected by cancer. And certainly we recognise that the best research innovation is enhanced through collaboration and pooling of resources and between people with complementary expertise. And to that end, the partnership between St. Vincent's and the Garvan Institute of Medical Research has been critical to demonstrate to certainly all of our patients that we have really the only integrated cancer centre in Sydney where the science is happening at the same time as the clinical care. And every day the scientists are seeing the patients and the doctors are thinking about the science in terms of how to help move the treatment forward, to help understand what's happening and to help make the next treatment even better. Now, our current approach at the moment practically relies on a few core pillars, some foundations of our approach is that we have a medical oncology research program, including clinical trials, to always try and bring the best treatments to patients, the best treatments globally to our front door. We have some infrastructure programs, which includes laboratories that we've set up to bring the best diagnostic tests to patients to help us understand their cancers. And we have some support programs for the younger investigators, including scholarship programs and grant programs to bring scientists and clinicians together with patients to push paradigms forward and to develop the next generation of clinical researchers. And certainly, I'm very proud to say that the visionary philanthropic support that we've received has catalysed and developed a number of careers and brought some very important advances forward on campus. And I could maybe just give you a couple of these just to speak to some work even beyond what Dr. Chin is doing to give you an idea of some of the projects that are at the cutting edge globally that we have at our front door. We have one of my colleagues, Dr. Barnett, is running a program called our Exceptional Responders Program, where we find those cancer patients like Theo that have had an amazing response to treatment, something we couldn't even have anticipated. And we try and understand why that's the case. So if we understand it at the genetic level, we can bring that innovation to future patients and to help further personalized care. And that's a national program that we're running in collaboration with some global partners from all around the world. One of my other colleagues, Dr. Liu, is running a program to help us understand why it is that patients can't get to the best trials on time, to understand the barriers to get patients who have no other options. Why can't we work together more as a team across Sydney and in fact New South Wales and in fact Australia to break down those barriers to get patients to the best treatments on clinical trials as fast as possible. And one of my colleagues, Dr. Crumbaker, is working on a trial in prostate cancer where we're turning the treatment paradigm on its head. So instead of taking away all the testosterone that feeds the cancer, we overwhelm the cancer with testosterone, and the cancer almost shatters into pieces. And we've had some very exciting early success with that approach as well. So these are 3 examples of some of the younger careers that have been certainly invigorated and by the visionary philanthropic support that we've had access to through the partnerships we've had both with the Curran Foundation as well as the Garvan Institute and Cure Cancer.
I love that image that you gave there of shattering the cancer into pieces. I think that's something that a lot of cancer patients would love to be able to channel during their therapy. It's an exciting time for cancer research right now. Looking to the future, Anthony, what do you see as the priorities to progress better outcomes for cancer patients and survivors?
So I think the key is to focus on what's important. Now, what's important is to help patients live longer and to help patients live better. And that's what we're focused on. And how do we do that? We do that through some very simple principles. One is we need the right infrastructure. So we need the laboratories and we need the clinical trials infrastructure in place. We need the right people. So every person we have on campus is contributing in some way. We think very hard about making sure we have all the support people in place that we need, the allied health people, et cetera, et cetera. We need the right mechanisms to bring these trials to patients. We need patients to be able to know they have access to holistic care, they have access to counselling, they have access to clinical trials. And we also need the right support from visionary donors to understand that actually what we need around here are a few angels, a few people that say, we're going to take a chance, we understand the vision, we understand the passion that these researchers have to help their patients, and to understand that the younger career investigators are those people that have that unbridled passion and enthusiasm that can really be the next breakthrough. So those are the I think those are the real principles that will help us shoot for the horizon, if you like, to really work on making tomorrow today.
Fantastic, thank you. Vanessa, let's go to you now. Can you tell us about the aims of your research project that's been enabled through this joint funding from Cure Cancer and the St. Vincent's Curran Foundation? What have you discovered so far?
So what we know is that patients with lung cancer who have immunotherapy, even though we have some indicators in sort of the routine tests that are done through the hospital of which patients may respond well to immunotherapy, those tests are far from perfect. And so when we're contemplating immune therapy at the start of treatment, it's really very difficult to tell who's going to do very well and who's not going to do well on immune therapy. So I work in the cellular sequencing lab at the Garvan Institute, where we have some very novel technology which enables us to take a cancer biopsy, pull apart all the cells that make up a cancer deposit, and we can actually look at the genome of every individual cell. And what we're trying to do is look at early markers of response when we expose these cancer cells and the other support cells to immune therapy in a petri dish. So practically what that means is when a patient at St. Vincent's is undergoing a biopsy for lung cancer, we run to the hospital, take some of the tissue that's taken out of the patient, run it back to the lab, and we plate it in 2 different petri dishes. One of them we expose to immune therapy and one we don't. And then after 4 hours, we then sequence all of those cells. And what we're trying to do is look at the very sort of intricate changes in the cell gene expression to see if we can identify early signs that that patient is developing an immune response to immune therapy. And what we've shown in our sort of early experiments, one is that this experiment is possible. This is quite a complicated experiment, but also that we can see, even after 4 hours, some changes in the T cell population that do indicate a response it has happened. And where we want to go from here is to see whether or not we can build up our body of evidence with lots of different patients to see if we can sort of see a spectrum there of patients that are clearly developing early signs of immune response and those who aren't. And would this be a more effective way of deciding for a patient, any given patient, whether or not they're likely to respond well to immune therapy? I think the other thing that we've really been able to show is that there are so many differences between each patient. And I think the current tests we have at the moment really are not giving us that ability to individualize patient decisions.
It's so exciting to hear about how you're effectively bringing the science laboratory like right to the patient's bed through this process. How would you describe the possible benefits to patients?
So look, my vision for this work is a little bit like when you have an infection and your doctor takes a sample of urine or blood and sends it to the lab, where a panel is run against different antibiotics to see what antibiotic suits you the best. So, you know, I think moving forward, could we do this for patients with cancer therapies? Could we take a specimen of their cancer in a petri dish and look at, you know, the standard immune therapies that we have now, but there are multiple different immune therapies that are becoming more available, and say, well, you know, this patient is highly likely to respond to immune therapy. This is the kind of treatment you should have, versus a patient who perhaps is not likely to respond to immune therapy, and a different approach could be taken. So could this be like a real-time functional marker of patients who will or will not respond to immune therapy? And so what we'd like to talk about in personalised medicine is giving the right treatment to the right patient at the right time. So we know that patients are the most well at diagnosis. We want to give them the best treatment up front. And so I think what we're really aiming for here is for better outcomes for patients.
It's such an exciting time for this type of research. And Vanessa, you've been a recipient of some of this early career research funding. What impact do you think it's had on your career and the research and the discoveries that you've been able to make? And what sort of impact do you think it's going to have on your research into the future as well?
Well, I think quite simply receiving funding like this enables me to do this research. Sadly, in Australia, government funding for early career researchers is very difficult to come by, and is particularly difficult to come by for people who have a novel idea for a project that really is very early on in its trajectory. So to try and get these early projects that are highly novel up and off the ground, they really aren't possible without funding like this. And so not only, you know, will we be able to see this particular project through, but by receiving funding at this point in my career, we can start to build on, you know, these projects that have been put in place for this particular project, but also to expand these translational projects for other patients with lung cancer. So it's really important, you know, it really is seed funding. We're planting the seed for this particular project, but hoping that a nice suite of lung cancer translational projects will build on our site.
I'd like to bring in someone who has benefited from all of this amazing research that's being done. Theo, that brings us to you and your experience as one of Vanessa's patients and a recipient of immunotherapy. Before we come to your treatment, though, can you tell us a little bit about your journey to diagnosis?
Yeah, thanks, Mary. Back in October 2017, I was at work as a physio and I went for lunch. And coming back to the surgery, I started to get dizzy and quite disorientated. And I sort of stood back and I thought, well, what's going on here? So I got back to work, continued treating, but I had trouble initiating speech. I had trouble using a phone, texting, writing medical reports. and I hadn't realised I just had a stroke. And the stroke was in the speech language part of my brain. And that occurred, I found out later, is because my blood with cancer was thickened. Anyway, so that night, I didn't say anything. That night I actually went to the football and I drove. And I was driving past all the suburbs and I thought something was wrong, and I'm trying to pronounce, I'm going Strathfield, Homebush, Rokerwood. I've gone, Rokerwood. That says Rookwood. And I thought, ooh, something's wrong. So I hid that again, and I didn't tell my wife when I finally got home. And lo and behold, 2 days later, I put my hand up and I ended up in St Vincent's. And they diagnosed me with MRI. I'd had a stroke. That resolved. And after about 5 days, so that was the good news. And the bad news was they couldn't find why I had the stroke. So they did a CT scan and lung cancer. I thought, oh, well, that's, not good. So prior to that, I'd been a smoker intermittently, on and off. I had my wife's laughing. I had a dry cough. I had shortness of breath. I would wheeze when I was asleep. I twice coughed up blood. I had a dream I had lung cancer. And being in the medical code, did I have cancer? Nah, don't be silly. All those symptoms were just unlucky. So I hid from that. But they did the CT scan, followed by PET scans, followed by lung biopsies of my lymph nodes. And they classified me stage 3C, which means I couldn't have surgery. So the next step, what do? I was sent to Professor Raj, a lovely guy, the head of radiotherapy at St Vincent's. And, I even got little tattoos of where they're going to do the radiotherapy. And so I was sent home, and the next day I was supposed to start chemo and radio, but he rang me that night and he said, I'm sorry, Theo. I said, I've been doing over and over and over, doing measurements, etc. And he said, well, it's too dangerous because where my lymph nodes had cancer, I'd probably end up paralyzed because they were very close to my brachial plexus under my arms. And anyway, so that was canned. So at this stage, the 6-month diagnosis of, life was sort of starting to hit me. I was starting to get my affairs in order. And, I had to tell my daughter who was in London. That was very, very difficult. She said, oh, well, that's the card you've been dealt. Let's get on with it and do the best. And so at that stage, no surgery, no chemo, no radio. But then I met Vanessa. So that's when the good news started. Can't hear you, I'm sorry.
Zoom meetings, eh?
Yeah.
What a great story. So the next part of the story where it really starts to get a lot more positive is when you underwent treatment with Keytruda, an immunotherapy drug under Dr. Chin's care. So can you tell us what your treatment experience was and why immunotherapy was so important in your case?
Well, because I had no other alternatives, Vanessa mentioned that there were some clinical trials being done with Keytruda, and we did a biopsy of my lymph nodes to get my PD-L1 level, which is a little switch on the cancer cells. And I had a very, 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 very high PD-L1 level, which made me a very good candidate to have immunotherapy, because the PD-L1 was, the way I understood, was like a switch that made my cancer invisible. So for the same reason that the cancer spread without my immune system fighting it, maybe this immunotherapy to get rid of the PD-L1 switch, expose it, and immunotherapy, the immune cells would attack. And it's a bit like Pac-Man. Have you ever seen Pac-Man, the game? I saw a video on iView about Ask the Doctor at one of the Melbourne hospitals, and you actually see the little cancer cells being eaten up by the little immune cells, and just killing them. And it was the greatest movie I've seen for a hell of a long time. So that was very exciting. So after that, we started with Keytruda, and that was every 3 weeks in Kinghorn. You know Kinghorn very well, Mary, in the Naloon Centre. All those lovely nurses looked after me. And I went there every 3 weeks for 2 years. That was good. You know, in the end, I was taking cakes and talking about football, and it was like it was quite nice to go there every 3 weeks and see all these people. I fainted once. They put a cannula in and they hit my vein, and the blood was spurting. And I don't like blood, so I fainted. Anyway, after we had CT scans every 3 months, and the lung tumour started to get smaller and smaller and smaller, and that was very, very encouraging. Vanessa was happy with the results. So where do we go from there? By March 2020, January 2020, I'd finished the treatment. The biopsies and the scans were all negative, but there was still a little bit of scar tissue, still a good 5 centimetre square in my lung. And we thought, with COVID about to close St Vincent's Hospital, it'd be a good idea to take the lung out, which we did that. And luckily we did. There was still some cancer cells within the lung, but there was a lot of T cells and lymphocytes that attacked the rest of the cancer, and they were forming all the scar tissue. So that was another good, medical decision by Vanessa and the team. So, that's about where we were with the treatment. And now it's been nearly 2 years since the treatment stopped. And initially I used to go in every 3 months, and I used to play a game with my wife. We'd sit there and I'd say, can you just see when Vanessa comes out with the results, can you tell if it's good or bad? And we'd look at this, and she'd come out, and the first few, the thumb was up before she even said hello. So that was good. But now the last few, especially my 4-year one, I feel that good. Vanessa rang with the results, and we talked for about 10 minutes about a holiday. And then I remember I said, oh, by the way, how are the results? So I knew they were going to be good, because she didn't jump in and say anything bad. So that was the immunotherapy. And it was very important, because I just, it was the only treatment, the only choice. I mean, I can imagine what happens now 4 years later. There'd be a lot more choices, but then it was, I was very lucky.
So Theo, tell me, how does that feel for you? You went from a position where it was like nothing was working, and you must have just had a whole series of consultations where it was just bad news every time you went in. And then suddenly it just got turned around, and it was all positive. How does that feel for you now? And how has that changed your own views on cancer research and care?
Okay, I had a running joke with Vanessa that I always thought if I lived 75 good years without being sick, I'd be happy. So I got to 60, and 60 out of 75 is a high credit. All right. Now, if I ever got a high credit university, I'd be excited. And Vanessa is now, we're at distinction level, and very, 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 very soon we're going to be high distinction, because I still feel pretty good. So, but about cancer research, to be serious, look, cancer, it causes, so many deaths. Cancer will grow, cancer will mutate, cancer will come back after remission, and cancer is very smart. Its whole role in the life of a cancer cell is to reproduce. But now we're getting some, really top-notch research doctors like Vanessa, who using their cutting-edge DNA, immunotherapy, single-cell sequencing, all these wonderful things, and they're starting to outsmart these cancers. And funding for this research is vitally important. Vanessa mentioned that the government doesn't give much. I saw a pie chart of, where all the money comes for funding, and the government's a bit low, but it comes from people like us who all can band together and help fight this. I mean, I walk into Kinghorn, and I'm humbled with all the names that are on the lists there, and then all the names that are in all these faceless people that that they list them in their gazettes and their journals and their etc. of all the donators, and and it's these people who helped save my life and many other people and yours, Mary, as well. All these wonderful people who did it because philanthropy. What does it mean? Friend of the people. So it's just great, and it's very very humbling. There's very few people who haven't been touched by someone who's actually had cancer, whether it's a family member, whether it's a friend, a work colleague. We all know. Like my father died in 1980 with lung cancer, and he lasted less than 6 months, and it was a really ugly death. And chemo in those days, it was a death sentence. And the research now, I'm 4 years, and my wife said the other day, I think you're healthier than you've ever actually been in the last 40 years. And that's to do with immunotherapy and the right mental approach. So I think we really need to help these researchers be smarter than cancer, and to provide better treatments. A cure for cancer is really no longer a dream. And if a donation today was going to save a life tomorrow, and you don't really know whose life you're going to save, because it could be a family member, it could be anyone. And these doctors, they need help, and I think we need to all join in. Thank you.
Yes. Yeah, some wonderful thoughts there, and I certainly share that sentiment that you had, Theo, there about all the gratitude that you feel when you walk into a place like the Kinghorn, and you see all the names of those donors.
When I see them now, I say thank you. It's all can you do? Yeah.
So I'd like to bring you in, Nikki, as St. Vincent's, the partnership between St. Vincent's and Cure Cancer has this enormous potential for supporting other early career cancer research projects. What drew you to the idea of partnering with St. Vincent's, and how would you like to see that partnership shape up over the next few years?
Yeah, thank you, Mary. I mean, for us at Cure Cancer, it's incredibly important. The partnership with both the Curran Foundation and St. Vincent's has led us to be able to fund Dr. Vanessa Chin's work for the next 3 years. And to Theo's point, over the last 54 years, we've funded over 500 early career cancer researchers. We exclusively do this because they don't get much funding at all. And this seed funding is vital in helping them have successes and building their career in cancer research. And to see the success Vanessa has had in treating Theo and many others like Theo is what keeps us going. At Cure Cancer, we fund all types of cancer research. Our tagline is "Cancer doesn't discriminate, and neither do we." And to enable us to do that and see these exciting results is incredibly important. At the moment, this year, the government has funded 5% of all applications of early career cancer researchers. That's very tiny. And unless we can continue to fund that pipeline across all areas of cancer research, there won't be any mid-career researchers or professors. So for us, doing what we do is vitally important. And as again Theo mentioned, people like Vanessa, although Vanessa is also a clinician, but a lot of our researchers just work in the labs. You would probably know the name of the Australian cricket captain, but you wouldn't know the name of the person that had put in place the cervical cancer vaccine in Brisbane. So for me, really, my focus is to talk about the wonderful work these researchers are doing, because they don't do it enough themselves. They don't have the ability to do that. So for me, this partnership, one, was incredibly important. Two, hearing from Theo and his results and the successes, that's just one example of the wonderful work the researchers we fund do. And that really empowers me, inspires me to keep raising money to fund these young researchers.
And across the sector, Nikki, what do you see as the critical factors for increasing the funding available for early career cancer research?
Yeah, it's very difficult because most people in the general public wouldn't know the challenges unless you're involved in the sector. You really don't know that there's a funding shortage across Australia. So for me, I really see my role as speaking out and letting people know, for showcasing the wonderful work these researchers do. We do that a lot in our newsletters we send out to our database, and Vanessa is just one of a cohort of over 500 we're very proud to talk about. But it's really just raising awareness that every step we take is a step closer to finding a cure. Often it's not a positive result, it's a negative, but that is also a step closer to finding a cure, because then you know you don't need to go down that pathway. So for me, it's about letting people know about the work the researchers are doing, how important and critical it is to fund early career researchers, and then celebrate the successes of the likes of Theo and many others around Australia and also globally that are testament to the wonderful work the researchers are doing.
Now, Nikki, we've got a question from our audience today. We're hoping that there are people out there who are keen to become donors to this fantastic research that's underway. So what tips do you have for people who want to donate to the most impactful cancer research? And how can they find out which projects are likely to have the biggest impact sooner rather than later?
Yeah, look, that's a very good question. Our current process is that we sort our applications and go through 2 levels of panel and peer reviews, and the research applications are then ranked on a basis of excellence. So as I mentioned, these could be any type of cancer research, including rare cancers, and they could be based anywhere in Australia as well. So we work with 32 of the institutes and universities around Australia. So on our website, we have the researchers that we're funding at the moment, and they are ranked in order of excellence. So any of those researchers we know are the top researchers in Australia at that time doing what they're doing. And obviously Vanessa is one of those as well. So for me, that work has already been done. It's a very stringent process, separate to Cure Cancer. So we don't have a say in that ranking. It's done independently. It takes 4 months to do. And then we have the privilege of just finding the money to fund them. Oh, sorry, you're on mute, Mary.
A question here for Anthony and Vanessa. Perhaps, Anthony, we could start with you. What role are cancer genomics playing in the treatment of patients now? And what do you think will be? How do you think this will be in the future?
So I think we're increasingly moving to a place where cancer genomics is going to be part of, in fact, it already is part of the workup for patients with lung cancer in particular, with melanoma, with colon cancer. We need to understand the engines that are making the cancer grow, so we have ways to attack them. So we need, and then beyond that, we have to understand the fuel. So we're increasingly getting more and more insights into the cancer, but we are at a critical point where there is a clear need for patients to have more access to cancer genomics to help tailor their treatment and personalize their treatment. We do a lot of that at the Kinghorn. We do a lot of research about that. But there's also a clear need for more discovery work to understand at that genetic level what is making the cancer grow. And then to integrate that with other treatments such as the immunotherapy to help patients who perhaps like Theo have lung cancer, but it hasn't actually responded to the immunotherapy. Can we add something on top of that that attacks the cancer, suffocates it, and then brings in the immune system to mop it up? So those are the things that we're working on actively with Vanessa's help and other researchers like herself at the Garvan, to understand how we can augment the understanding of the genetics of the cancer with other treatments to further improve cure rates.
Vanessa, would you like to address that as well?
Yeah, so I think it's, we already are using genomics in the diagnosis and treatment of lung cancer. So there are several genetic tests that are done routinely on newly diagnosed patients to look for some very specific mutations which link with specific therapies. And what we've always known is that wider, more general genetic panels that can be run on patients with lung cancer are likely to pick up those rarer mutations, which also link with therapies. Now, up until very recently, those wider genetic panels have only been available to patients either through a research program or if they choose to pay out of pocket for a commercial test. At St Vincent's, more recently, there's been a sort of extended panel of 50 genes that has been applied to patients with new diagnoses. So we are picking up some of these rarer genetically mutated lung cancers, which can link up with the therapy. Now, where the field is moving is now there are some genes that are known to predict for poorer response to immune therapy in lung cancer. So that's something we are picking up now with these extended panels. And where I think we are going to be moving forward is having genetic information not only on the cancer, but on the non-cancer cells. So genetic information on the immune cells, looking at immune cell activation in response to immune therapy, but also people who get really, really extensive immune activation and toxicity. So I think, the field is moving forward in the clinical space where genomics is being used more and more. And then the research that I'm doing is using sort of more novel techniques to look at lots and lots of data on a given single patient. But even in the time of Theo's treatment, if he was to be diagnosed today, we would actually have more genomic tests done on his tumour as a matter of course than we had available to us back in 2017.
Another question here for Anthony. Where does genetic counselling fit in with the types of research projects that are being discussed today?
Well, thanks for the question. So obviously genetic counselling is very relevant to some patients with cancer in general. And what we have to separate is the changes in the cancer versus the changes that the patients have been born with. And they're two separate things. So when you've got a significant family history of cancer, then certainly we can come in and do some tests to see, were you born with anything that would predispose you to getting cancer? And now we can answer the question, do we have drugs for those things to treat your cancer? So, for example, with my prostate cancer patients, I'm always asking them the question, do you have a family history of cancer in your family? And if so, I want you to get tested for some of these genes that cause prostate cancer, because now I can tell them that the genetic counselor will tell them that not only is it important for the next generation, but it's actually important for them. Because if the genetic counselor finds those genes, then I can prescribe drugs to help treat their cancer. So it is becoming increasingly critical that the patients understand that we are testing not only their cancer sometimes, but also testing to see if they were born with anything that gave them cancer, because now with modern technology, we have drugs we can use to attack the cancer with that knowledge.
Maybe you could just put another question to both Anthony and Vanessa, because you're both heavily involved in the research space, but also clinically. As clinicians who are facing patients every day, how have things changed for you in that sense that you now feel like you have more tools that you can reach for, more therapies, and that sense that with ongoing research, there are going to be even more therapies available for some of your patients? Maybe Vanessa, we could start with you.
Yeah, look, it's a really exciting time to be a lung cancer oncologist. I guess when I was doing my training, we had a relatively narrow suite of treatments for patients with lung cancer. And now we do really have a menu of different treatments that are suitable for different patients. So it is really exciting. It can be slightly overwhelming for patients, I have to say. We tend to order a lot of tests. We have a lot of complex discussions around genetics and genomics and clinical trials, but ultimately what it means is that with the right sort of counselling and discussions with the patient, that we can hopefully arrive at a treatment that is best for their cancer, but also best for them. And I think, patients are really well educated on this stuff. So I'm constantly surprised. Most of my patients, before I've met them, have done some of their own research, and they're expecting me to talk to them about genomics. They're expecting to talk to me about clinical trials, and they're expected to be sort of presented with different treatment options. So I think that, we sometimes, as doctors, I think, downplay the research that patients are now able to do for themselves. So it's really nice to have some options to give patients, I have to say. It's a lovely problem to have too many options.
And your thoughts on that, Anthony?
I mean, I certainly echo those sentiments. Personally, I've been, for example, been treating melanoma now for 20 years. And when I started, there was literally nothing that we could do in someone who has had melanoma that spread. But now I use the word cure in the consultation, which is an amazing transformation within my lifetime, that I can use the word cure in a consultation in a patient with cancer. So these are the incredible changes that are happening at an incredible pace every day. And we are very privileged to be at the coalface of that transformation.
I think I can remember exactly where I was standing on which day when one of my doctors used the word curative, and it does give you. It's one of those words that when you hear it and you know it, and it's a great word to hear when you're talking to your clinicians. Nikki, just a final question for you. There's so much medical research out there. There's so many frontiers in the medical science. How do you choose where the funding goes? We've got genomics, we've got immunotherapy. Those are really the exciting frontiers at the moment. Your thoughts on priorities for the future.
Yeah, look, for us, we don't choose. The researchers come to us and apply. We are the only charity focused exclusively on funding early career researchers. So within the research sphere, I think it's safe to say most researchers know who Cure Cancer is and what we do. So the researchers will come to us and they'll apply for a grant. We give one-year grants for 100,000 or two for 200,000, and then we go through the excellence ranking that I talked to you about before. The reason we have decided to keep our sphere and scope so broad is that cancer changes as we find cures, as we find ways to treat cancer and prolong life of people that are going through cancer treatment. We're also seeing the emergence of rare cancers as well, based on a lot of other circumstances, like not only your genetics, but environmental factors, etc. So for us, always being on the pulse of evolving scientific research is incredibly important. We've recently had a breakthrough with pancreatic cancer. There's a survival rate over 5 years of only 8% in pancreatic cancer. And that's using a drug that already exists now to treat pancreatic cancer. So for us, our work is never done. But I love what I do. It's an absolute privilege and an honor to be able to fund researchers like Vanessa and to hear stories like Theo and get results. And we have, and we've done that for the last 54 years, and I want to keep doing it for another 54 years. It's an honor. And, we're very, very focused on finding a cure for the different cancers.
I think that's a fantastic sentiment to end this on. Can I thank all of the members of the panel who've joined us here today for a fascinating discussion. And Richard, I'll hand it back to you.
And thank you for your time, for joining us from South Africa, and for facilitating so really professionally today's discussions. And can I also thank the panel of Anthony, Vanessa, Nikki, and Theo. Thank you for sharing those fascinating insights that have so clearly highlighted the importance of supporting early career cancer research. As medical research and new treatments continue to evolve at such a rapid pace, we have to honour the commitment of all of our supporters that are helping St Vincent stay at the leading edge. Your generous support ensures that the best possible patient care and treatments are available to all St Vincent's Hospital patients when they need it the most. For that, we're most grateful. Thank you all on behalf of our hospitals and the Foundation. If you'd like to support St Vincent's by making a gift, and Theo quite clearly indicated how important philanthropy is, you can visit www.supportstvincents.com.au. That brings tonight's webinar to a conclusion. Thank you all for joining us, and a very good evening to everyone.